Hello, and welcome to another episode of the Science on the Edge podcast. I'm Dr. Mark Darcy, and in today's episode, I'm going to talk about stem cells, embryonal stem cells, adult stem cells, and induced pluripotent stem cells. So let's get started. Okay, so stem cells and differentiation. So first of all, what are stem cells? Well, they are a kind of unspecialized cell. They can be found in embryos. They can be found in adult organisms also. I'm going to start by talking more about the embryonal stem cells, and I'll move on to adult stem cells a little later. So in the embryo, they are unspecialized cells that eventually develop into many different specialized cells. The process by which a cell becomes specialized is differentiation. This basically works by a cell switching on and off various different genes. A single cell, single fertilized egg, is unspecialized. So the egg and the sperm come together. You have a fertilized egg known as a zygote, and that zygote has what we call total potential. The cells can become anything. They can become brain, skin, muscle, heart, liver, kidney, whatever. They are unspecialized. But then they begin to differentiate. As the cell divides, becomes a ball of cells, and eventually an embryo, a fetus, and an individual. Different cells switch on and off different genes. So in humans, we have about 23,000 genes, but different genes are switched on in a skin cell than a heart cell, for example. That's why your skin cells don't contract. So the process is known as differentiation, and all multicellular organisms have some form of stem cell. In animals, stem cells are found in the embryo, where they differentiate into all the cells needed to form a fetus. In terms of properties of stem cells, they have the ability to self-renew, so they can divide and produce more stem cells. Usually a stem cell divides into two. The daughter cell differentiates into some type of cell, say a skin cell, and the parent cell remains a stem cell. Differentiation, the ability to develop into these specialized cells, so they can renew themselves and they can differentiate. They're also found, as well as in embryos, in adult tissue, where they differentiate into specialized cells that are needed to replace damaged tissue. So, for example, your skin cells are constantly dying. They rise to the surface of your body and then they're shed. So new skin cells need to be reproduced constantly. So we have stem cells, unipotent stem cells, I'll talk about potencies later, in our skin that give rise to our skin cells. Also in our intestine is constantly being damaged by the food and the chemicals in our gut. So the cells die and new cells need to be produced. In our liver too, a lot of turnover of cells. Some parts of the body have less stem cells, so brain, heart, hardly any, as far as we're aware, because those types of tissue don't have the same turnover that maybe the liver and the skin does. So stem cells in the bone marrow, which are a type of multipotent stem cell, can differentiate into red blood cells, the erythrocytes, and white blood cells, the neutrophils. But all sorts of different tissues are produced by stem cells, even in the adults. As for the discovery of stem cells, they were originally discovered, as the name suggests, in the stems of plants. So plants are always growing, so stem cells are needed to make new shoots and roots throughout their lives. Stem cells are found in the cambium part of the plant. In the root and stem cells of the vascular cambium divide and differentiate to become the xylem and the phloem. The vascular cambium forms rings inside the root and shoots. Cells divide and grow out of the ring, differentiating as they move away from the cambium. Now, in terms of types of stem cells, embryonal stem cells I've already talked about. They're derived from the inner cell mass of a developing blastocyst. So that's an early embryo, and I'll talk about that a little more in a while. So this is about 5 days after initial fertilization. So the egg and the sperm come together, give rise to these totipotent stem cells, well, stem cell, the zygote, and it divides into a ball of cells known as a morula. And each of those cells is also totipotent, can give rise to any type of tissue. Then about 5 days or so after conception, the first set of differentiation steps occurs, and you end up with something called a blastocyst. Blastocyst has an outer ring of cells, which give rise to the placenta, and an inner cell mass, which gives rise to every other type of cell in the body. So adult stem cells, these stem cells are present in adult tissues, such as bone marrow, discussed earlier, adipose tissue, fat cells, and the brain in smaller amounts. Adult stem cells are multipotent, meaning they can differentiate into several cell types, but are limited to the tissue they reside in. And some adult stem cells are unipotent. They can give rise to only one type of cell, such as the epidermal stem cells. Finally, we have the induced pluripotent stem cells, the iPSCs. These stem cells are generated by reprogramming adult cells, such as a skin cell, using various genetic manipulation techniques. These iPSCs are similar to embryonal stem cells in their ability to differentiate into any cell type in the body, which might make you think, well, that's easy. Why bother with embryonal stem cells? We can just use those. It's more tricky than it sounds. There are issues, which I will get into shortly. There are also fetal stem cells. These stem cells are present in the fetus, so early in development this is, and can differentiate into many specialized cell types. There's cord blood stem cells. These stem cells are collected from the umbilical cord and the placenta of a newborn baby and can differentiate into various specialized cell types. Cord blood stem cells are used in the treatment of blood disorders and immune system diseases. Now, this is an interesting one. You've probably heard of this. Some people are offered the ability by companies to actually freeze cells from the umbilical cord of their children following birth for future use. Considered this myself when we had our child. Does cost a little money to do because these cells need to be stored pretty much forever, but they have such potential they could possibly be used later on in the child's life to treat various health conditions, perhaps in ways we don't even understand yet. There's mesenchymal stem cells. These stem cells are found in the bone marrow and can differentiate into bone, cartilage, and fat cells. Mesenchymal stem cells also have anti-inflammatory properties and can help repair damaged tissues. When it comes to potency, I've mentioned totipotent stem cells. These are cells that have the ability to produce all cell types, including all specialized cells in an organism and the extra embryonic cells, which basically means the placenta and the umbilical cord. Then the cells start to differentiate. At the blastocyst stage, the inner cell mass are what we know as pluripotent stem cells. They have the ability to produce all the specialized cells in an organism, but not the placenta or the umbilical cord. In humans, totipotent stem cells are only present in early life, the embryo. They differentiate very quickly at the blastocyst stage and continue to do so after birth. So here is a blastocyst, 5-day-old ball of cells, less than the size of a full stop. The outer ring of cells here is already differentiated and will give rise to the placenta and the umbilical cord. This inner cell mass here will give rise to every organ and tissue in the body. So these embryonal stem cells come from the inner cell mass of the blastocyst, and they can differentiate into any kind of cell. They're pluripotent and have many potential uses. So when it comes to embryonal stem cell research and clinical use, you start off with one of these blastocysts, so 5 days old, and then you harvest the inner cell mass. Very delicate process. You then plate these cells out on a petri dish, and use various chemicals and techniques to get them to differentiate into all types of cells in the body, from neurons in the brain to hepatocytes in the liver to myocytes in the heart, etc., etc. It's very difficult to produce different types of cells this way, but we're learning gradually to produce every type of cell this way, which offers the potential to treat pretty much any disease. Someone has a stroke, part of the brain is now dead. It doesn't come back. Perhaps by implanting neuronal stem cells into the brain, that is, embryonal stem cells, which have been differentiated to become precursors of brain cells, we could repair the brain tissue. The same goes for the heart. You have a heart attack, part of the heart is now dead. Perhaps we could differentiate stem cells, and this has already been done, to become heart cells. And then maybe we could implant them into the damaged heart and they would repair it. In theory, we could use this technique to treat pretty much anything from baldness to blindness to deafness, missing teeth, Crohn's disease, diabetes. This is an interesting one with diabetes, actually. This has already been done clinically. So you take stem cells, you differentiate them to become beta cells. Those are the cells that make insulin. In type 1 diabetes, those cells are dead. So the individual cannot make their own insulin and they need to take insulin injections every day. Well, in the lab at least, we have created beta cells from stem cells, which can make insulin. They can then be implanted and have been implanted in mice and have successfully produced insulin and cured type 1 diabetes. In theory, pretty much any organ or tissue in the body could be repaired using stem cells. But this doesn't just apply to embryonal stem cells. When it comes to adult stem cells, there are clinical uses too. So here's a procedure that's already been tried. So an individual is blind in one eye due to severe damage to the cornea, the outer layer of the eye. But the eyes do contain stem cells, a few unipotent stem cells that can make cornea. So this is a process. One, you remove a tiny sample of stem cells from the good eye. You then grow them on a contact lens for 10 days with the right nutrients, of course, in an incubator. Then you place the contact lens in the damaged eye for 3 weeks or over the wound. The stem cells realize where they are, heal the damage to the cornea, and sight quickly improves. So this technique has already been tried successfully. Now that's just one technique. There's many more techniques in the pipeline. Now let's talk briefly about induced pluripotent stem cells and the Yamanaka factors. So adult cells can no longer differentiate. They've lost that ability, which is probably good, because if they could, we would probably be more susceptible to cancer, because they could differentiate into all types of tissue and perhaps spread through the body. However, there is potential use. There are issues with embryonal stem cells. They could be rejected by the host if they're transplanted into another individual. The immune system of the recipient could reject the cells, but not if the stem cells were produced from the individual's own adult cells. And this has now been done. So the discovery of the 4 Yamanaka factors, OCT3/4, SOX2, K1F4, and c-Myc in 2006 was a major breakthrough in induced pluripotent stem cell technology. These genes, initially identified by Dr. Shinya Yamanaka and his team, have the ability to induce pluripotency in somatic cells. So basically, these genes make proteins, and these proteins, they reset a cell back to factory settings, back to that pluripotent stage. It's understandable that we would need these factors, these transcription factors, because obviously when you produce an egg or a sperm, well, those cells, when they fuse together, need to result in a totipotent zygote. Everything needs to be reset so that a new baby, a new organism can be created. So it makes sense that these factors exist. It's such a small number of factors as well. So Yamanaka has basically given us the ability to rewind the clock when it comes to adult cells and turn them back into stem cells. In fact, this has even been done in mice, adult mice, rather than just taking a cell out and turning it back into a zygote and then rolling it forward, making an embryo, taking the cells out and using them for medicine, which will be done and is being tested right now. What has also been tried is to actually add these factors in very small amounts in mice and rewind the clock of an old mouse. Its cells regenerate, become younger, and the mouse becomes younger. There are issues. If you switch on these factors for too long, the mice develop tumors. It's a very delicate process, not ready to be used in humans yet, but great potential. But at the moment, these induced pluripotent stem cells can, in theory, be used to basically create new cells, tissues, organs from an individual's adult cells, and then give them back to that individual to repair damaged tissue, cells, and organs, perhaps indefinitely, increasing longevity by a huge amount. So let's go through this process. So the process of producing iPSCs typically involves the following steps: isolation of adult cells. Adult cells are obtained from a patient through a biopsy or blood sample. Introduction of the reprogramming factors, so the Yamanaka factors. They can be introduced into the cell using a virus or by other means. Culturing the cells, so growing them in petri dishes. They're cultured and monitored and allowed to grow and divide. Then there's selection of the iPSCs. The reprogramming process is not always efficient, and the resulting cell population will consist of a mix of different iPSCs and partially reprogrammed cells. Scientists need to identify the good ones, the ones they want, isolate them, separate them, and use other techniques and factors to get them to differentiate into the type of cell needed. Now, there are ethical issues to all of this, quite a few ethical issues. First of all, when stem cell research began and started to make some headway, George Bush Jr. in America was president, and he decided to ban all government funding of embryonal stem cell research. Bush is a very religious man, and he had various religious advisors, and he was advised that you cannot fund this research because you're taking one life away to save another. Basically, the idea was, according to Christianity, that as soon as the egg and the sperm fuse together, the soul enters the body, the moment of conception, and therefore you're destroying a life to save another. So throughout his presidency, funding in the US stopped. Continued in the rest of the world, though, which is one of the reasons in Cambridge, in the UK, where I live, we have quite a few stem cell labs that are doing quite well. Wasn't banned here. And when Obama became president, the ban was lifted. Although in America, it's still pretty dangerous to work on stem cells. You need armed guards outside the buildings because, well, in America, people get a bit crazy about these things, let's just say. Okay, but there are ethical issues nonetheless. Other than that, this isn't I personally don't believe in the soul, so this one doesn't bother me, but there are other ethical issues. For example, we could end up in a future, if this is an expensive therapy, where only the richer people can afford it, poor people can't. Imagine a future where richer people, or at least richer parts of the world, these individuals get to live much, much longer, perhaps centuries. The body just being topped up with new stem cells every now and then, damaged organs and tissues repaired, either using embryos or adult stem cells that have been rewound back to the stem cell stage. Richer people could live centuries, be young, be fit, be healthy for centuries, whereas poor people might not be able to afford any of this and would live much shorter lives. This would divide the world even more than it's divided already, and could allow the rich to get even richer, to accumulate more power, and to hang around for a long time. That is an ethical concern. Some people have suggested even banning this sort of research because of that. But the problem is, if they did try to ban it, I mean, there would just be clinics opening up in places like Cuba or wherever, and rich people would go there. So banning it wouldn't help. What we need to do is make it more available and cheaper. But technologies do tend to get cheaper over time, so this is a possibility. This is a possibility. The other issue that people bring up is overpopulation. Overpopulation could be an issue. The planet's already overpopulated, and this could add to that. Certainly things to think about. Science does not work in a vacuum. But I will say right now that already huge progress has been made. Scientists have already grown an entire heart using stem cells. We have a shortage of organs for donation at the moment. That could end soon. People are working on treating pretty much every disease with stem cells, and we're at the clinical trial stage with many of these techniques. For example, Parkinson's disease is a disease caused by the death of dopamine-producing cells in the brain, and that causes serious neurological issues. Now, we have made dopamine-producing cells from stem cells in the lab already, and they have already been implanted into the brains of people with Parkinson's disease. We're waiting to see the results. Many diseases, from dementia, Alzheimer's, Parkinson's, to heart disease, to diabetes, to muscular dystrophy, blindness, deafness, Crohn's disease, pretty much any disease could be treatable with stem cells in the near future. It's an amazing time to live. So regeneration may be our future. We may be living in a future not too far from now, where we have the possibility, when our body wears out, when it gets old, to roll it back again and have another life and choose when we die. Brave new world. Okay, thank you very much for listening. If you want to know more about me, you can follow me on Twitter. My handle is @MarkOfTheD. But thanks again for now. I look forward to speaking with you again next time.